Connections between neurons. Connection between neural assemblies. Connections between areas and regions. Connection between the brain sections. Global connections in networks. Brain activity pathways are made of several dynamic intermediates and global connectivity with a different frequency range of dynamics. The brain networks are organized in multiple spatiotemporal scale layers. Their topological changes in different scales, and when activated expresses the encoded information as a memory unit. They range from individual local networks to global networks. Consolidation and fine convolution affects resonation of two radial and circular strain energy streams and short-term strain streaming residual production, as well as long-term memory. What kinematics of the brain activities describe the cortex convolutions? Long-term memory motif structuring is generally according to short-term memory components of localized, layouts, setup, and dynamic, as well as intensity definitions. At the same time, shared components will be consolidated in the same location as a long-term memory motif. Combination of these two factors induces different strain forces, especially over the cortex inner layers that support different convolution of the cortex layers, depending on usages of the same long-term memory motifs by the distribution of them based on consolidation strengths. In humans, convolution is much more intensive because of multi-times more short-term memory forming and conversion to long-term memory. Long-term memory consolidation and cortex layers convolution are rooted in molecular deformation and connectivity configuration deformations. In waking time, short or long fibers will return feedback of produced concepts to over the parietal cortex, where in subconscious states, unprocessed concepts, the formed short-term memory, will recycle back to the sensory brain. Association fibers interconnecting cortical regions in one hemisphere with adjacent or distant regions of the same hemisphere are categorized as short association fibers, arcuate fibers, or long association fibers. The long association fibers may have numerous fiber systems entering, exiting, and traversing them. Important named bundles include the uncinate fasciculus, the superior longitudinal fasciculus, the superior and inferior occipitofrontal fasciculi, and the cingulum. The cingulum is a bundle through which the major monoamines, dopamine, norepinephrine, serotonin, and part of the cholinergic projections travel to their widespread target sites. Thalamocortical radiations are the nerve fibers between the thalamus and the cerebral cortex. Functionally, thalamocortical fibers radiate energy from the thalamus to the cortex, relay sensory or motor information from the thalamus to the cerebral cortex through its nuclei. A schematic diagram of the connections between thalamic relay nuclei and their corresponding cortical areas, of the same color, through the thalamic reticular nucleus. Black lines indicate corticothalamic connections, and colored lines indicate thalamocortical connections. A. Anterior thalamic nucleus, M. Medial thalamic nucleus, VA. Ventral anterior nucleus, VL. Ventral lateral nucleus, VP. Ventral posterior nucleus, LP. Lateral posterior nucleus, PU. Pulvinar, C. Centromedial nucleus, P. Parafascicular nucleus, LGN. Lateral geniculate nucleus, MGN. Medial geniculate nucleus, TRN. Thalamic reticular nucleus. Thalamus, as center of radial inputting to the strain carrier wave center round the cortex. A strain-saturated cortex, too much releasing strain residual, as well as too slow or repeating sensory input make thalamus to submit less radial inputs to the cortex middle layer. As the result, assembly oscillation and circular strain flow through layers are not in balance, slow circular kinetics, and the brain phase reduces to sleep condition. These changes are caused by the thalamus and its interactions with its inputs and the cortex. When the thalamus allows less input through, the subject becomes more drowsy or enters deeper sleep. In short, with the less radial bombardment of the cortex, strain carrier waves will cool down. As he falls asleep, the gate slowly closes off progressively more channels of input. In performing this function, the thalamus also interacts with modulatory systems. Modulation is memory consolidating by restoration between circular strain circulation frequency through and vertical oscillation in an assembly. Neural assemblies form by resonation of assembly oscillation with carrier wave cycling frequencies. Joining of recalled strain packages from neural assemblies in coagulation will form a short-term memory current over the sensory or a working memory over the motor brain, as well as segregated theme and emotion of that current memory in the limbic system. Thalamus is a switchboard that will put strain packages in order in the current memory. The switch relays in the thalamus that activate related cortex areas. Radial, static, strain package bombardment from the thalamus will excite neural assemblies over the cortex through their middle layers. These signals also excite the motor brain and receive inputs from the sensory brain through the limbic or straight, forming weaker working memories. The hippocampal formation and the Papez circuit are responsible for strain carrier waves theme signification and refining emotion to reduce irregularity in streamlines. Especially the 3 layers of Ka and 3 layers of Dg are involved in this dissociation. In the cortex area sequence of activation, hippocampal and Papez's loops have the main role. Consequently, the inputting signals should be ordered in the same way, and activation of the limbic system components as well, which they have also an order of strain signal transferring within the limbic system, activation starting from the hippocampus to the fornix and so on. Direction of area activation over the cortex. Direction of component activation within the limbic system. Sequence of the thalamus inputting lateral segments. Sequence of the thalamus outputting lateral segments. Where the thalamus sends information signals to the cortex middle layer 4, it receives back information from the cortex through the cortex layer 6. Specific thalamus relay nuclei relay information from a specific sensory modality to its corresponding cortical area. The LGN, for example, receives input from the optic tract and sends its output to the primary visual cortex. Association nuclei receive input from specific areas and project to somewhat generalized association areas. Nonspecific nuclei both receive from and project to diffuse or nonspecific regions. Cells in the thalamus project to layer 4 of the cortices. Pyramidal cells in layer 6 connect back to the thalamus in much greater numbers. In fact, the ratio of feedback to projection fibers is nearly 10:1. Thalamus has a central role in the brain in activation of sensory areas over the cortex in sequence of activation of its related part and as the result, the direction of circular strain carrier wave round the cortex. The sending to feedback receiving ratio indicates how much potential strain energy within neural assemblies will convert to kinetic strain energy round the cortices as strain carrier wave. The sequence of different primary sensory areas, especially somatosensory, visual, auditory, is so important in coagulation of strain packages in the lateral parietal cortex and increase in formed short-term memory that will enter the hippocampal formation and the limbic system in general. Somatosensory and sensory-like information as recycles from the prefrontal cortex have the least kinetics level. Next to the occipital cortex and visual primary area, the above-mentioned stationary strain packages will be boosted by entry visual information and form short-term memory information to perceive the collection. It is further will boost by entering primary auditory information. By the above explanation, the thalamus segregates and relays information so that the strain carrier wave circulates in a certain direction. By weakening that segregation due to less activity, the strain carrier wave kinetics will reduce and the brain state drops from focus to relax waking, and further to sleep stages. Separation of inputs, including conceptual inputs. The different nuclei of the thalamus all preserve a separation of inputs. When input into the thalamus is topographically organized, the thalamus even maintains uneven magnification present elsewhere in the brain. Not only sensory inputs, but also conceptual inputs communicate through the thalamus through the mediodorsal nucleus of the thalamus, MDN, and the thalamic reticular nucleus, TRN. Thalamus. The diagram of communication, schematic diagram of the connections between the prefrontal cortex, PFC, mediodorsal nucleus of the thalamus, MDN, and the thalamic reticular nucleus, TRN. In left. The role of pumping strain energy and paying attention. Polarization of the thalamus sections and related neurons in a harmonic way in concentration, paying attention, and depolarization in relaxing and sleepy brain states will back the pumping strain energy role of the thalamus in the brain. When relay cells are slightly depolarized, they fire in what is called relay mode. In this mode, there exists a linear relationship between the stimulus to a relay cell and its response. Information is thus passed on reliably to its destination. In contrast, when the relay cells are slightly hyperpolarized, they fire in burst mode, a cluster of firing followed by an unresponsive period, even though the stimulus continues. The information thus is not transmitted faithfully. To select an item for processing from a noisy environment, resonation between strain carrier wave and entering input is required. The thalamus acts as a strain energy pump round the cortices. The brain state depends in its activity degree. Its reduction in activity is related to degree of strain residuals accumulated over the cortex neural assemblies, which play as resistance force against its pumping characteristics. Then the brain efficiency decreases, and it feels sleepy. The power of paying attention and the type of inputs to pay attention depends on the thalamus activity. The direction strain energy pumping round the cortices is not only based on sending sensory information to the cortex areas. The maximum feedback information from the motor area and decreasing strength in the sensory brain with an increasing gradient through the prefrontal cortex will accelerate strain carrier wave circulation. The most evidence for the above is almost no motor signal in the sleep state. Sensory, motor, and limbic inputs synapse onto both interneurons and the proximal dendrites of relay cells. They are, in general, excitatory, utilizing glutamate. The feedback from the cortex, discussed above, is also glutamate-based and thus excitatory. It contacts interneurons and distal portions of relay cells. The feedback from the reticular nucleus, on the other hand, is inhibitory, using GABA, and synapses on proximal dendrites of relay cells. The location of its synapses suggests that it modulates sensory input. The brainstem also has its say, sending inputs to relay cells, interneurons, and the reticular nucleus from cholinergic, noradrenergic, and serotonergic systems. The gating function of the thalamus depends on these connections. Recall that activity of 10 Hz is associated with drowsiness. If the thalamus is isolated, it will show the same 10 Hz activity. The modulatory systems in the brainstem act on the reticular nucleus, which in turn acts upon the relay cells, eventually producing 10 Hz firing by hyperpolarizing those relay cells. The result is a certain distortion of thalamic inputs. Olfactory association area function dual, both radial and circular, streams. Olfactory is the only sensory input which does not pass through the thalamus to the cortex. Olfactory, other than basic sensory functioning, promotes instinctual motive to strain carrier waves to inspire. The olfactory cortex is the portion of the cerebral cortex concerned with the sense of smell. It is part of the cerebrum. This system is involved in the processing of our emotions, survival instincts, and memory formation and connects senses, such as odors, to our memories and emotions. The cortex areas overstraining by inputs through thalamus and its emotion arousal cause. Thalamus is closely in connection with the Papez circuit, which segregates not regulated strain packages in short-term and working memory currents. The cerebral cortex exchanges radial information both with the thalamus and the entorhinal cortex. The earlier form radial strain streaming exchanges. The latter initiates circular strain carrier waves, that contain strain packets, around the cortex. The thalamus as the gate of inputting strain radial signals to the neural assemblies over the cortex submit strain streams by resonation. Resonation of oscillating assemblies in the same time, meaning within the same fragment of one cycle around the cortex, will happen with a specific frequency that matches with strain carrier wave cycling's frequency on one side. On the other side, the thalamus submitting frequency will resonate with specific neural assembly oscillations. Thus, the thalamus functions as a filtering gate for inputs that strain carrier wave receives. Filtration is accompanied by activation of certain assemblies in one fragment as feature integration. An assembly oscillation depends on dynamic strain residual changes within it. In this article, I will argue that the thalamus exchange of radial signaling between the cortex and the limbic system energy loop of Papez will be focused. Anterior nucleus of thalamus is the gate between the Papez loop and the cortex strain carries energy attractor. Importance of this gate is essential for all brain processes, including sensation, perception, feeling, and cognition. The thalamus consists of a pair of egg-shaped cell groups at the top of and continuous with the brain stem, see above figure. It is extremely important for perception, as evidenced by the fact that 98% of all sensory information is relayed through it. In fact, olfaction is the only sensory information that does not pass through the thalamus on its way to the cortex. Specialized portions of the thalamus relay the information from each sensory modality separately. Association nuclei also integrate the different inputs. Emotional internal inputs will be collected from the Papez circuit through mammalian to the thalamus. Other than sensory inputs, the thalamus also receives non-sensory input, conceptual, from areas important in motor control and emotion, namely the. Finally, subcortical nuclei create a closed loop, generating feedback to the thalamus. The reticular nucleus is of this type. Note that its inhibitory cells create an extremely fast, negative feedback loop. Cerebellum, basal ganglia, and limbic system. Obviously, it must be performing some important function. Non-sensory inputs are releasing strain residuals from different brain areas, known as conceptual inputs or internal intermediate products. They include the processing products of sensation, perception, feeling, and cognition, as well as motor signals. They are considered as feedback or recycling inputs that will be sent back spread to the cortex areas. The prevailing hypothesis holds that the thalamus functions as a sort of gate or filter, deciding what input, if any, of the multiple channels to allow through for processing. As any gate can, the thalamus can allow all, none, or some of the information through. This hypothesis was applied in a recent study of schizophrenics which found that the thalamus of a schizophrenic patient was significantly smaller than normal patients. The researchers proposed that schizophrenics, due to the irregularity in the thalamus, cannot close the gate to ignore useless or irrelevant information. Increase in thalamus signal frequency will change the brain state to wakefulness. A demonstration of the thalamus EEG recordings in waking time shows that have the characteristics of small amplitude, high frequency, 20 to 80 Hz, activity. But in a sleepy state, the thalamus is characterized by high amplitude, lower frequency, 7 to 13 Hz, rhythmic activity. By falling into deeper sleep, the frequency of activity decreases while its amplitude increases. Reversely, when the subject reaches higher wave frequency in REM, sleep, the submitting frequency approaches wakefulness again. Limiting the thalamus signal submissions to releasing strain residuals takes the brain into a sleepy state and radial bombardment of the cortex. Consequently, strain carrier wave kinetics will reduce. As the result, the thalamus will become effective in a memory unit building in local and next connection modulatory aspects. In memory unit building, neural assemblies oscillate with specific frequency that resonate with slow cycling strain carrier waves. When such a resonation matches an assembly connectivity oscillating frequency, a specific thalamus part will resonate with that specific neural assembly oscillations, and the thalamus filtering gate will be set for that input. In other words, in the future any similar input makes the thalamus activate a certain memory unit. Filtration is accompanied by activation of certain assemblies in one fragment as feature integration. The thalamus' primary function is as a relay center through which sensory inputs exchange signals between the spinal cord and brainstem, as well as produce conceptual inputs to the cerebral cortex. Receiving sensory and conceptual inputs, by the thalamus, will be tuned for related neural assembly over the cerebral cortex to determine the location, type, and duration of sensation, perception, and cognitive as well as motor actions. Strain carrier waves process current information, short-term and working, by their interaction with the long-term memories over the cortex association areas. However, this information have frequent different processing overall types by being transferred through different tracts. Tracts connect cortex association areas together. In this processing, the cortex activated locations by the inputs from the thalamus, brainstem as well as other cortex areas are highly influential. Both the hippocampus and the amygdala among other the limbic system component are effective in activation of the cortex locations. A representation of the limbic system and tractography of its main pathways are shown on the right. Among the brain's global neural networks that are the main loops, which conduct strain carrier waves over the cortex, and their interaction with inputs, mainly from the limbic system, some long connections are an icon. The main connections of the hippocampal diencephalic and parahippocampal retrosplenial network are the ventral cingulum, the fornix, and the mammillothalamic tract. The end stations of this network are indicated in yellow. The main nodes of the temporo-amygdalo-orbitofrontal network, indicated in green, are connected by the uncinate fasciculus. The dorsal cingulum is the main connection of the medial default network, whose cortical projections are shown in blue. The connections between amygdala, orbitofrontal cortex, and posterior occipital areas. The inferior longitudinal fasciculus and inferior fronto-occipital fasciculus. Pyramidal neurons conduct radial signals to the cortex. The cell bodies and dendrites, in red, of 3 pyramidal cells in the cerebral cortex compared with the intracortical distribution of axons, in blue, arising from these cells. Axon collaterals distribute over a much wider area than do the dendrites arising from the same cell. Elementary processes in selected thalamic and cortical subsystems, the structural substrates. Scheibelme, Scheibelab, 1970. Number of pyramidal tracks to / from the cortex locations reflects the number of neural assembly groups that each may be a hub connected of some local networks. Pyramidal neurons in each neural assembly connect layers networks, from inner to outer and reverse. Long pyramidal tracks and between cortex layer pyramidal neurons are in continuation of each other and facilitate input-output, radial, exchange of strain signals. Pyramid versus granular and other neural connections between the cortex layers in neural assembly scale. Samples of receptor distribution in different areas of the cortex. These illustrations indicate how specific neural assemblies in different cortex areas may receive specific radial information from inputs. Activation of each assembly will decode a specific memory unit in the location to be integrated with information from other assemblies along a pathway to generate a thought or other brain products. Higher ion channel numbers in layers 2/3 make stronger horizontal energy streams, and reduced ion channel numbers in layers 5/6 make stronger vertical energy streams within neural assemblies. Dendrites integrate subunit information in groups of receptors in units. For humans, as the size of neurons increases, the density of channels found in the neurons also increases in outer cortex layers to keep kinetic strain energy high. This may be more limited in inner cortex layers to reduce kinetics and increase the potential to dispatch to distance receptors in the limbic system and the brainstem, as well as reconfiguration of connections in inner layers within neural assemblies. However, the distribution of ion channels in pyramidal neurons differs on the axon side compared to the number of ion channels in dendritic spines. Further to that, it depends on the distance from the cell body. In mammals before humans, the gradient is smoother, and interaction between outer layer activity, current memory, and inner layers, location for long-term memory units, is less active. Organization and local connectivity of M1 pyramidal neurons. M1 contains 3 groups of excitatory pyramidal neurons: intratelencephalic neuron, IT, pyramidal tract neuron, PT, and corticothalamic neuron, CT. Excitatory input to IT and PT neurons arise from inter- and intralaminar connections, shown as broken arrowed lines. Layer 5 point and IT neurons receive strong interlaminar excitatory connections from layer 2 or 3 neurons, shown as gray squares. Intralaminar monosynaptic connectivity comprises recurrent connectivity, IT to IT and PT to PT, in addition to unidirectional connections from IT neurons to PT neurons. There are reciprocal inhibitory connections. Radial information input/output to and from the cortex, radial exchange of information between cortex layers. Pyramidal tracts are bundles of nerve fibers that originate from the brain's cerebral cortex and relay signals to the brainstem or the spinal cord. Pyramidal neurons are found in association or memory areas including the cerebral cortex, the hippocampus, and the amygdala. Examples of these connections are the primary excitation units of especially in the prefrontal cortex and the corticospinal tracts. They also connect different cortex layers in a location. Outer layer pyramid neurons direct information to inner layers to consolidate memory. Inner layers pyramid neurons direct information to outer layers to recall. Number of neural radial and horizontal connections over the cortex indicates number of neural assemblies that each encode an information. What makes human different is huge number of memory units it can build to encode information, and ability to recall them. Human brain is differentiated from other mammals for higher number and more dense neural networks, where it is increased many for him in the evolution tree. It seems by increasing number of neurons, possible number of connections, ion channels, also increases. However, it has found that this is not true for humans, especially in inner cortex layers. Indeed, it is the reverse in humans. The comparison for pyramid neurons in layer V indicates a significant reduction in dendritic ion channels. Where pyramid neurons are responsible to convey input messages to the cortex layer 4, and output messages from layer 6, the formatting messages will transfer from layers V and 6. Formatted message from these layers will conduct to outer layers, a special I/O, and 3 to integrate into the strain carrier waves that travel through these outer layers. In reverse, the high kinetic strain waves through outer layers will induce strain packages in the neural assemblies to format in the inner layers memory units. Huge number and finer memory unit sizes provide possibility to combine them in many different ways. Decrease in number of horizontal connection, where vertically through pyramid axons remains high, indicates how the number of memory units can be increased and size of memory units tend to be smaller sizes to encode finer and more precise information. In other words, how more detailed memory units can form for humans. This is the significant difference between human and previous mammals that make human unique. Summary: knowing structural connections in the brain as connectome, conducting information through activated connectivity groups as connectivity, and combination of holding information in connectome, traveling of information, connectivity, under functional continuum, is the basis of the kinematics of the brain activity theory. Do not forget to send your comments and questions to moz.dini@gmail.com.